Everyone. My name is Jeff Harden. I'm from the Department of Integrative Biology, and I'm a trainer in the genetics PhD program. And it's my privilege to be hosting our speaker for today, Dr. Mark Peifer. I've known Mark for a very long time, and he asked me to keep my biographical comments short, so I'll try to do that. After his undergraduate work, Mark did post-bac work at the University of Minnesota, where he really got the itch to do bench science, in particular genetics. After that, he did his PhD with Welcome Bender at Harvard, working on the bithorax complex, a classic complex that you may know about, contains homeotic genes, and graduated in 1988 with his PhD. From there, he did postdoctoral work with Eric Wieschaus at Princeton, initially working on extradentical, I think. He's nodding, that's good. Very important for the activity of transcription factors in flies, particularly the homeotic complex. And then where I know him, his work the best is because he discovered a very important protein in Drosophila by identifying the molecular identity of a mutant called armadillo, which turns out to encode beta-catenin, very important kind of double agent in the cell involved in Wnt signaling, wingless signaling in the case of fruit flies, but also cell-cell adhesion. After that kind of groundbreaking work, Mark, not surprisingly, got a great job at UNC Chapel Hill, where he has been in the Department of Biology ever since. And he came to UNC in 1992. After coming to UNC, he did continue to do amazing work on armadillo and associates. We also did work on other proteins that are near and dear to my lab's heart. One of them is a protein called ENA, which is an actin regulator. And I'm sure we'll hear a lot about another protein that his lab has worked very extensively on today, a protein called canu in Drosophila. It's called a fadin in other organisms. Mark is a leader in cell biology. He is on the editorial board of Journal of Cell Biology, Journal of Cell Science. His stamp is everywhere in the field of at the interface between cell and developmental biology. Since 2006, Mark has been the Michael Hooker Distinguished Professor of Biology at UNC. And on top of all of the research that he's done, I think one of the things I particularly admire about Mark is that he thinks about students very carefully and puts a lot of energy into students at the undergrad and the graduate levels. He's been involved in an REU program at Chapel Hill for a number of years. And I think the genetics students who are going to interact with him in the roundtable are in for a treat. Mark's going to tell us today about building the body plan, the miracle of morphogenesis. Please welcome Mark.
Okay, thanks. As Jeff said, we met a long time ago when we were the honorary C. elegans and Drosophila people in a particular area of cell biology. That's changed, but it's been great to see him again and come back to my Minnesota roots and get my butt frozen off. Just to point out, that's what my campus looks like right now. Okay, so hello. No, what are you not going to? There we go. Ever since I started work as a PhD student, I've been interested in what I think is really one of the fundamental questions in biology, right? How does a single cell fertilized egg, right, self-assemble into the body plan of an animal? And that's my now 32-year-old daughter, right, a few years ago, right? It's a miracle, right? No outside input, right? And out comes this incredibly complex animal. And as Jeff said, we thought about that in a variety of different ways, but thinking for many years about the Wnt signaling pathway, which I'm not going to talk at all about, but also really thinking about how you literally physically assemble the body, how you build body, tissues and organs, how cells change shape and move. And it's cool, right? But it's even cooler when you watch it happen, right? So this is a fruit fly embryo, gastrulating, something that you did, that I did, that all animals almost, I learned from Phil, not his guys, do, right? And again, in Drosophila, in just a matter of an hour and a half, you're going to turn a hollow ball of cells filled with yolk into something that's basically the animal, right? It's going to have an elongated anterior posterior body axis, a narrowed dorsal ventral body axis because it's convergently elongated. What we couldn't see in this view is a group of cells going in like the neural tube goes in a vertebrate embryo, except in Drosophila, they're going to form the mesoderm. You would have seen cells here apically constricted. Cells over here would also be apically constricted to make that gut tube that's going to run in between. Again, it's remarkable, right? How do cells do that? And you can even see in this still, which we're basically looking at the actin cytoskeleton, right? Cells that are already in the midst of some of these rearrangements, shortening some of these borders to form a group of cells like those guys in the rosette, right? So how do they do that? And again, it was fun and exciting to be part of work done by groups around the world, right? That really built a baseline model for how cells link to one another and how they use the actin and myosin cytoskeleton to change shape, right? With the idea that, again, transmembrane cadherin molecules bind to cadherins on the neighboring cell, again, lots of them around the apical circumference. Cytoplasmic domain binds to beta-catenin, binds to alpha-catenin, binds to an actin filament. So contracting actin filament can change the shape of the cell. And that was great. We were really proud of it. Lots of work from lots of different labs. The problem is that it's wrong, right? So many people, but important work was done by James Nelson, by my grad school classmate Bill Weiss, by Alex Dunn, right? To show us that this model is oversimplified in two really important ways, right? One is, and we'll focus a lot on that there's a lot more proteins in this space, right? That this idea that it's a simple linear chain linking the cytoskeleton to junctions is inaccurate. And second, that this whole machine, and that's an important part of what Bill and James and Alex did, is a mechanosensor and it's mechanoresponsive. And in simple, what they showed was while the interaction of actin filaments, the alpha-catenin in this three-protein complex is inherently relatively weak, if you touch an actin filament, and Alex can do this with his laser trap, right? If you touch an actin filament to that three-protein complex and pull it in the right direction, in his case toward the slow-growing end of an actin filament, suddenly this entire machine turns into a tight actin binder, right? And again, he often Alex often now shows you that people still have those little Chinese finger traps, right? Again, you pull on it and the tension gets tighter. And again, what we saw happen in gastrulation, all those cell shape changes and movements, right? It makes sense that you might evolve a system, right, that strengthen connections when you pull on them. And in fact, our colleagues working on integrin-based junctions were way ahead of us, right? People like my neighbor, Keith Burge, because they already knew that integrins were linked to the actin cytoskeleton by a complex of probably 100 to 120 proteins, right? Including proteins with a wide variety of different roles. And again, that this whole machine in the integrin situation was mechanoresponsive, right? So really as cell adhesion folks were really catching up with them. And so our overall goal, right, as a field, and particularly my own goal, right, is to really understand how this happens and to understand how it happens at every level of organization. I'm interested in and have been since my first lab rotation in the structure of proteins at the atomic level. And then taking that all the way up to how does the animal gastrulate, right? Really trying to understand that at every level. How do cells change shape and move, link junctions to cytoskeleton, and do that to generate a lot of force, but not rip the animal apart, right? That's really our challenge. And before I talk about science, I want to spend a minute, and it's interesting that Jeff introduced me that way, right? To talk about something that really has changed in my lab in the last 10 years, which is postbacks have become an essential piece of my lab. All of these folks were postbacks in my lab in that time period, right? Many of them in a shorter version of that. And it's really transformed the way that we do science. And it's also really fun. It's so much fun to have young people in the lab, but also fun from a PI's perspective of not having to make a 5, 6, maybe 7-year commitment to a PhD student, right? And really bring people together in teams. And again, if somebody wants to ask me about that later, talk about how one of my own science heroes, Sandy Schmidt, got me started down this road. But these folks have been really critical to the work that we've done. And there's a whole nother project, a set of projects going on in my lab that I won't touch on, right? That were initiated by a just remarkable postdoc, Mike Bishop, and being continued by postdocs Art Clark and another postdoc, Siobhan, and some other younger folks working in the lab, right? To study how cells collectively migrate. This is a system that Mike developed as a PhD student where muscle cell precursors are going to enclose the testis in a sheath of muscles. And it's a fantastic story. And again, if anybody's interested in that, I'd be happy to talk to them about it. But now let's go back to that junctional cadherin-catena complex and its complexity. So again, the original model had this linear connection, E-cadherin, armadillo beta-catenin, alpha-catenin, actin. But we now know that there's probably dozens of proteins in this space, and that every one of them is multivalent. That's going to be key, right? They're all binding to multiple partners, right? And as we're going to see, all or almost all of them do that using both folded protein domains and intrinsically disordered domains. And so it's a network, not a linear linkage. And I'm going to try to convince, and again, I want to understand how that works. I'm going to try to convince you that the place to take this apart is by studying the protein that Jeff mentioned, the protein that himself is called canoe and other animals is called the fatin. Why? The first is because it's not as important as E-cadherin or the catenins. If you take those guys away, as soon as gastrulation starts, the embryo falls apart, right? It basically becomes a puddle of cells. And you can't study how cells change shape and move when they aren't linked to one another. Other proteins in this network, like a juba or a sidekick, I can have a vial of flies that are null mutant. This is the C. elegans thing, even for things like ENO, which is embryonic lethal in Drosophila, right? Where they're fine, but if we look closely, we see defects in cells changing shape and moving. But canoe, and we also sometimes work on Z01 polyketoid, right? Sit in a sweet space in that because canoe is important, and this is work over multiple years in my lab and other labs, for just about every place where we link junctions to the cytoskeleton. The first place is when you actually assemble that ring of cadherins up at the apical network. So these are cadherin puncta that should all be localized here, apical to basal, should be all localized in that junctional zone. And the canoe mutant, they go all along the lateral membrane. Again, I told you the cells ventrally are going to apically constrict like neural tube cells, right, to fold in to form a tube. They do that with an apical contractile network of myosin in green that should cover the entire apical surface of the cell and be linked to those red junctions. And in a canoe mutant, the cytoskeleton continues to constrict, but it rips off the junctions and they fail to fully fold in the epidermis. That intercalation we saw that elongated the germ band involves planar polarized localization of myosin along the anterior posterior borders. We should only see a single line of myosin because at this resolution, the myosin on the two sides of the junction should be a single line. But again, in the canoe mutant, it's pulling apart and that elongation fails. And it's called canoe because at one of the last stages of morphogenesis in Drosophila, the skin, which is only covering the ventral and lateral surfaces, needs to migrate to the dorsal midline and zip closed. And if it doesn't do that, the guts fall out, the skin ends up shaping like this, and it looks like a canoe. We have better names. I'm sorry, we just have better names. How does canoe do that? It's part of this mechanosensor, right? So if we use an antibody to canoe, right, what we see is it's present in junctional puncta, biomolecular condensates all around the circumference, but it's especially enriched at tricellular junctions and at a later stage at those aligned anterior posterior borders where the myosin was enriched. So anywhere where you're pulling hard on junctions, canoe gets recruited there. And work from Jen Zollin's lab showed that it actually is force that's doing that, because if you treat the embryos with a ROCK inhibitor to reduce myosin activity, that enrichment goes away. And then if you take away canoe, right, in those places where cells are being pulled on most vigorously, right, so during the interpolation, again, it's going to be centers of rosettes or aligned anterior posterior borders. Again, we're using the Mercury junctions. Those junctions pull apart. And that will be the end of germ end elongation. And again, a canoe mutant's embryonic lethal with defects in many of the important morphogenetic movements that the animal has to undergo. So that's one reason. It's critically important for all the places where we link junctions to the cytoskeleton. But the second reason is because it's a complicated protein. And almost all of the junctional proteins are complicated proteins. You know, again, I was educated in a different time where proteins had domains and often a single domain, right? Like a kinase domain. What we now know is that proteins often have many domains and also have regions that aren't structured. And canoe is particularly complicated. It has 5 folded protein domains with some shorter intrinsically disordered regions, and then 1,000 amino acid intrinsically disordered region. And so that complexity also is something that will help us understand how you build this incredibly complex and important machine, right? And all of that is well conserved in across the animal kingdom, right? Including in sponges, right? But this is just contrasting the fly and the human protein, right? All those folded domains, 40 to 75% identical, right? So one thing I have been interested in ever since I started my career, and particularly since I started my lab, and in fact, one of my first graduate students did exactly the same sort of thing to beta-catenin, is I want to understand how that machine works, right? Why does it have those parts, and what do each of those parts do? Because we won't understand how gastrulation works until we understand how the machines work. So we've got to take the machine apart, right? And that's what we're going to do. We're going to take it apart systematically. And that work was initiated, that part of the work was initiated by a now former graduate student, Kia Perez Valle, tremendous addition to my lab and really changed shape of what a bunch of the people in my lab do, right? And again, she wanted to know how the machine worked and she wanted to take it apart. And at that point, we thought we knew how the machine worked, right? Because we knew from work that my collaborator, Kevin Slepp, had done along with my graduate student, Jessica Sawyer, that the PBZ domain can grab a hold of the C-terminal end of E-cadherin or another junctional protein, transmembrane junctional protein in flies called mechanoid and mammals called nectins. We also knew from work that the Takai lab initially did and then Jessica Sawyer did in my lab that a region in the C-terminal half of what we now call the intrinsically disordered region could physically bind to actin filaments in a filament pull down, right? So it grabs a hold of junctions by binding E-cadherin and it grabs hold of actin and it's a linker. And it's regulated, like many other proteins in the cell, by binding the small GTPase, in the case of canoe, the small GTPase RAP1. We were pretty sure it bound this region. Again, we ultimately did some biochemistry, Leah did some biochemistry to define that more carefully. But again, the idea was RAP1 binds, activates canoe. We know some things about what that means. There are other things that we still don't understand, and most of my original models turned out to be wrong. But that activates canoe, grabs E-cadherin, it grabs actin, and it strengthens that connection. That was the hypothesis. And so, again, we've got amazing tools in Drosophila, right? wasn't trivial, but Kia re-engineered the canoe locus, right, at the locus, right? So we put into the locus any version of canoe that we want, put in a wild type version that's GFP tagged, or delete those 3 domains that we thought were the key pieces, right? The key working parts. If it can't be activated, because we deleted the RA domains, it can't bind BCAT heron because we deleted the PDZ domain, can't bind actin because we deleted this so-called F-actin binding region, right? It'll be dead, right? So it should have been pretty simple. The problem was that hypothesis was completely wrong, right? So again, she generated the animals lacking the PDZ domain, the animals lacking the FAB region. There were multiple generations out, right? All the proteins they have lack those domains, and it's a viable adult, right? I will just say right now, because this is published work, right? If you look closely, things go wrong. You don't even have to look that closely with these guys. With that stock, 25% of the embryos die, right? And natural selection will easily remove that from the system. This guy, we had to look actually quite a bit more closely to see things going wrong, but that's what we saw, right? So individual connections, even though the protein's essential for embryonic development and morphogenesis, individual connections that we thought were going to be critical aren't important, right? Again, we have these more sensitized assays. Now, in contrast, the RA domains were important, almost essential, right? So when she deleted those, again, the animals were embryonic lethal, right? And their phenotypes, we assess them in a whole bunch of ways. I'm just going to show you one example, right? Their phenotypes are consistent with them being almost, but not completely null, right? And again, here's just one example, right, of that canoe delta RA mutant, right, with gaps forming, right, at the centers of vertices and at these aligned anterior posterior borders. Everything we do has to, I mean, it's the modern world. Everything's quantified. There's statistics, da 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 da. But again, this is published work. Strong, but not completely null. Another thing we learned from this is none of those 3 regions is essential for localizing canoe to junctions. All of those mutants go to junctions. 2 of them go to junctions in that tension-dependent way. This guy doesn't, right? So the RA domains are also important for the enhancement of localization in response to mechanical force. So that was where we were at in 2021, right? And subsequently, the lab has done a lot, right? Again, some of it, right? That's a first author. That's a paper where all 3 of those authors, including the first author, are postbacks. There's another paper where all 4 of those authors are postbacks, the first author included, right? There's Kia's paper, right? But Christie actually was postback. We've now deleted individually all of the folded domains, right? Every single one of them. And the only one that's important is the RA1 domain. If you delete the RA1 domain, it's basically like deleting both RA domains. And again, Leah showed that both of those domains, unlike what the literature suggested, both of them bind RAP1. They bind RAP1 with very similar affinities within 2-fold of one another, but only one of them is essential. This one's actually not even published yet, but it's true that that's also, all of those guys are viable lines. Again, if we look closely, all of them have defect. Okay, so all of the functional domains are individually dispensable. And that degree of robustness is not unique to canoe, right? Many of the other junctional proteins are similar, right? So here's some work from one of my lab alums in his own lab, Tony Harris, working on the protein in flies called bazooka and most animals called par3. Again, complex multi-domain protein, big intrinsically disordered regions. And Tony found, right, that you could delete every individual domain in bazooka and the protein still was recruited to junction. Here's some nice work from Uli Tepes. Even alpha-ketinin, which again is essential, right, has domains that are dispensable, right? So this idea of robustness is not unique. Why, again, all we can do is speculate about why questions, but because it's really important that this works, right? If you screw up morphogenesis, right, that's not a good thing. And in my mind, the evolution has over-engineered the system, right? So it's got lots of backup plans, right? So these individual domain deletions, which might reflect what might happen in kind of stochastic fluctuations that happen during real development, right, are still accommodated. Okay, but so we, again, with the exception of RA1, none of the folded domains are essential. So what is important? Well, we still got almost half of the protein, right? This long, intrinsically disordered region. Again, something that's shared by Drosophila canoe, human defadin, by the homologs in at least most other animals. So what are these intrinsically disordered regions? Well, they remain, but especially it was a few years ago, right? One of the hottest topics in cell biology, because again, we've all been so focused on the important roles that, folded domains play in enzymatic activity or protein interactions, right? But suddenly people realized, right, that almost every protein has a region that is intrinsically not structured, right? And that we better figure out what they do. And they fed into this idea of, and I'll say it, but I don't religiously believe in it, that proteins can phase separate, right, like oil and vinegar, right, and we can form organelles that aren't bounded by membrane, right, that these IDRs are key to that. And they're doing that by relatively low affinity interactions, but that are highly multivalent, right? We'll talk about stickers and spacers, right? But there are regions that maybe are higher affinity binding sites, regions that are both providing length extension, but also probably lower affinity interactions. And by having dozens of different multivalent interactions, right, individual molecules can form into large multi-protein complexes. And adherence junctions are biomolecular condensates, right? So again, Tony Harris, back when he was a postdoc in my lab, was one of the first people to use confocal microscopy to what was then the limit of its resolution, and could show something that Uli had shown with EM years before, that junctions are punctate complexes, and Tony in his own lab went on to show that every one of those dots contains 1,500 plus or minus 250 cadherons, right? He did molecule counting. That's also true in mammalian cells. And that's a picture from our lab, and other labs have done that. I'm really interested in this right now. Again, another project in my lab is try to understand what's inside that dot once you get past that limit of resolution of a standard confocal microscope, and we already know some things. So Anya Schmidt found that even though in my Macintosh diagram, canoe is right there in that complex with cat heron and armadillo, that armadillo beta-catenin in red and canoe in green are enriched in different adjacent puncta, right? So there's probably structure inside those. And again, Rennick is one of the people pursuing that, and probably now in collaboration with your Grossons in Germany. So coming back to, again, sort of how people think about IDRs today, right? At least many people think of them as divided conceptually into what they call stickers and spacers. Stickers are short regions that, at least in some cases, are acting as binding sites for folded domains of other proteins, right? In our case, we're going to see binding sites for F-actin filaments, right? And the spacers are, again, literally being spacers, but also probably interacting in lower affinity ways, right? To help build things into these complexes. And many junctional proteins, most junctional proteins probably contain intrinsically disordered regions. And in an in vitro assay, many or most of them can phase separate. And this is a nice paper last year from the Tagashi lab where they showed that a Fadden mammalian canoe, right, is one of those proteins that can phase separate in vitro. Right. So what's going on with that intrinsically disordered domain? And again, that both canoe and a Fadden have one, yeah, but they're very different from one another, right? They're different in length, right? The Fadden one is about 100 amino acids shorter. They're different in amino acid composition. Often these IDRs are enriched in particular amino acids, sometimes homopolymeric rungs, right? They have different amino acids enriched in them. They're different in how many positive and negatively charged residues they have. And if you, outside of this C-terminal region called the FAB, where we already knew there was about 30% identity between the two of them, you take the rest of the IDR of one of those proteins and blast the genome of the other animal, right? There's no significant similarity, right? So that's, again, you know, when we got protein folded domains that are 40 to 75% identical, that sounds very different. But along came this incredible tool that's really changing how we think about things, AlphaFold. And AlphaFold can not only tell me almost certainly the structure of all the folded domains in my protein. And again, my collaborator, Kevin Slepp, and the postdocs working in his lab have really made use of that. We'll see a little bit of that, right? But they can also look at the IDR and ask, are there any regions of the IDR that have short regions that might fold, right? And so, again, I could say we did that. In other words, we went to the already existing database that they've created of all Drosophila proteins, right? And we looked at both and all human proteins. We looked at both canoe and a Fadden. And what we found was most of the IDR in AlphaFold is unstructured still, but there were short regions that were alpha helical. That C-terminal 30% identical FAB region, right, has multiple helices predicted that are shared between the two. And then there are other helices that are present either in the mammalian protein, in the fly protein, or in both. One example that I think is important for Jeff and I talked about this for their work is that the vertebrate protein has a helix that has got a known function. That helix binds to what's called the M domain of alpha-ketinin, right? That helix is completely absent in canoe, right? Absolutely clear that it's just not there, right? So again, some of these things are evolving independently in different lineages, right? But some of the two of the other helices, discounting the ones in the FAB, were conserved, right? And once we saw the helices, we could go back to comparing sequence, and now you could see that there's a region that's about 30% identical spanning these two alpha helices, right? So that allowed us to conceptually divide the intrinsically disordered region into stickers and spacers, right? Two conserved, potentially helical regions, and the FAB and the central part of the IDR, which we're going to call H and FAB, and then the extremely not conserved regions, amino terminal and C terminal for the central helices, which we're going to call N and C, right? So again, don't know anything functional, but that's what we did. And again, we had already deleted this region, and that was one of the mutants that was viable. So a team of people, a postdoc, Corbin Jensen, and three postdocs, Noah Gurley, Yufei Zhao, and Adrian Matthias, decided to start taking apart the intrinsically disordered region to try to figure out what, does it play a role, right? Because again, the rest of the folded domains, only RA1 was essential. And they started by doing something, the most extreme thing, which is just to delete all of the rest of the IDR that's not the FAB. Okay. And in doing all of this work, I want to emphasize, right, that we're doing using these crazy cool genetic tools and pushing the microscopy and cell biology to the limit, but all of it's done in parallel with Kevin Slepp, right, and a series of postdocs in his lab. But the person who's relevant for this is Leah Wolfsberg, right? And again, Kevin's the one who literally solved, using crystallography, the structure of the PBZ domain in collaboration with us. Worked with a different postdoc, Amber Butcher, to model the structure of the dilute domain and show that it's in part acting as a dimerization machine. And then Leah had measured the affinity of each of the RA domains to RAP1 and Kevin had modeled them, right? So all of the work is going to be done in that parallel way. But first, we're going to bring in really our simplest assays. Our simplest assays are if we alter or delete a region, is the animal dead, right? And that turns out to be the most powerful assay, right? If it's alive, it retains substantial function. And then we do something even almost as simple, which is we look at what's called the cuticle. We look at the skin of the animal. This is how that original Bichat-Smith-Volhard screen that changed the development of biology forever was done, right? You take the egg of an embryo, you take an embryo at the end of embryogenesis in its egg, and you dissolve everything but the outer skin, which is also their skeleton, in a drop of acid, and you can see the larva inside, right? A little overexposed here, but right, head at the head end, tail at the tail end, and you can see morphogenesis work. And again, canoe, complete loss of function disrupts a process called head involution. It disrupts that process that I told you about, dorsal closure. So you end up with this skin shape like a boat, which also has holes in it because it also disrupts epidermal integrity in the cells that are doing the most cell shape changes, right? The ventral epidermis. And so this is a canoe null mutant, right? Maternal and zygotic. And most of the animal, almost all of the animals have a failure of head involution and dorsal closure. And most of them also have big holes in the cuticle, right? So that's the no function phenotype. Deleting the proximal IDR what was pretty severe, right? The embryos died, but they didn't live to be adults. They died during embryonic development 100% of the time. They had defects in many of the morphogenetic movements, but as you can see with the blue bars moving this direction, right? It's not null, right? So that protein retains at least some residual function, which would surprise. Again, if you look at morphogenesis, right? The things that canoe is important for often fail, right? So the ventral furrow invagination should have closed the ventral furrow. Here's a guy with an open ventral furrow. Frequency isn't as strong as the null mutant, but those things go wrong. There are gaps at aligned anterior posterior borders or tricellular junctions, but they're much less frequent and less severe than in the null mutant, right? So again, this protein is pretty debilitated, but it's not completely debilitated. And again, the big scale morphogenetic movements like dorsal closure often fail, right? So deleting the entire proximal IDR substantially reduces, but doesn't eliminate function, right? But we got another surprise when we did this, right? When we look at wild type canoe, it's at junctions. And as I told you, every other one of the mutants that we looked at, it was still at junctions. RA domain, not at enriched at under tension, but it was at junctions. When we delete the proximal IDR, it's still at junctions, but at much reduced levels. Again, and there's quantification that was done. But the crazy surprise was instead, almost all the protein was in the nucleus, right? So green there is canoe IDR, should be localized here in these apical junctions. There's a little bit in the apical junctions. Again, we saw that when we looked at them from this, from this side on. You can already see the nuclear localization starting to appear there because it's slightly curved. Most of the protein was in the nucleus. And initially we're like, oh, I hope this isn't just some weird fly thing. But then again, the Higashi lab published this paper. They never mentioned it. But if you look in almost every one of their figures in the places where they deleted the proximal IDR, right, there's the protein accumulating. There are reports that Afadin does things in nuclei. And there have been many reports that it can localize to nuclei. So it wasn't entirely surprising. We had never seen it. And I wasn't entirely happy about this because I've been there before, right? I went to work on, well, actually, I went to work on armadillo thinking it was going to be the receptor for the Wnt ligand, right? Which it did not turn out to be. But, here's this protein that I could be working on in junctions, and it's in the nucleus, and it's doing a really important thing there as a transcriptional coactivator, right? So there's some collaborative work with the Peifer lab on that. But there are also proteins that we've worked on that people claimed went into the nucleus that in fact we never had seen in the nucleus until we blocked nuclear export. That's what Vera did in this paper, right? And damned if this Wnt regulator APC doesn't go in the nucleus. It does if you block nuclear export. But what Dave did was tether it to a variety of places outside of the nucleus, including a transmembrane domain in the mitochondrial membrane, and you can completely rescue its role in insulin. So again, the fact that it goes to the nucleus doesn't guarantee that it's doing anything there, right? But we'd never seen wild type in the nucleus. But Mike and Sarah, because the postdoc working on this had gone to leave for another job, I begged them to do what's actually a pretty difficult experiment, which is to permeabilize the eggshell of embryos and bathe them in a well-known nuclear export inhibitor called leptomycin, right? So that if the protein goes into the nucleus, it'll get trapped there. And when they did that, wild type canoe, there's a control without permeabilized, no leptomycin treatment, canoes at junctions. And there it is with leptomycin treatment, wild type canoe is going in and out of the nuclei. Okay. So we're still thinking about that, right? And also doing experiments to try to address whether it needs to go into the nucleus. We had earlier faced this, right? So one of my first graduate students, Rich Cox, right, generated a mutant of beta-catenin that had the lipid modification signal of RAS attached to its C-terminus. And what she found was that mutant could completely rescue the role that beta-catenin has in cell junctions and was completely deficient in rescuing its role in Wnt signaling. Again, it had to go into the nucleus, right? So we now have generated both CAX-tagged versions of canoe and guys with a meristylation sequence at the amino terminus, right? And are testing them for function. Okay, so stay tuned for whether it has to go into the nucleus. I hope not. I don't want to work on transcription. I really don't want to work on transcription, but you follow the science where it goes. Okay. So that said, the idea was important. And we again, it used to be if you showed that a protein phase separated, you could publish a paper in Nature, right? There was that brief window, right? And Cliff Brandwein and some other people became very famous based on that, right? But now we actually have to figure out what they do. And there actually is not that much information. There's a fair amount of information. What happens if I take it away? I heard some nice work from the Bement lab yesterday, right, about that, right? But taking it apart is not something that's been done a lot. And most of that's been done with the guys who are the RNA in the RNA things like the genome granules, right? So very little work on how different parts of the IDR work, right? And again, we start with this idea that that Afadin and Canu's IDRs are quite different, right? They're quite different from one another. Outside of those two stickers, right? No sequence similarity, right? So we were pretty sure that that one wasn't going to replace the other, right? So again, we did this simple experiment, right? Put the ephedrine IDR in place of the canoe IDR. We also put the canoe IDR back into the canoe because there were little things that happen at the two junctions that we wanted to account for. And damned if they both rescue, right? And again, if we look hard at the ephedrine mutants, there are small gaps. If we look at the eye, which is one of our other places that go-to places to look at the pupil eye, where again, flies have these amazing multi-ommatidial eyes, where each ommatidia has an exact number of exactly this number of each of these types of cells, and they all have their own cell shapes and had to gain them by cell shape changes and rearrangements, right? We see defects. And in fact, this guy is a little bit of accumulation in the nucleus. So its nuclear export signal is probably not completely compatible with the fly one, but you get a viable fly. And the ephedrine guy, again, if we look just at junctions, right, brings canoe back to the junctions pretty well. It is not as sensitive to tension as the wild type protein. Okay. So now we come back to this idea, right, that we could take the idea apart, RDR apart, and it's got two regions of helical conservation, right? One called H and one called the FAB, even though no one had ever asked whether that region binds actin, by the way. That's another long story about how that short region got to be called the FAB, even though all of the experiments were done with a region about that big. But those were the two regions, right? And again, despite its name, nobody had ever measured that. So again, along into this project come Kevin and Leah, right? And they can ask, are those regions that bind actin? And it's a relatively simple, I mean, again, even I might be able to do this maybe, right? You buy actin, you add the right buffer conditions and it polymerizes into filaments. Filaments are big. You put them in an ultracentrifuge tube, you spin, and the filaments pellet. And if something binds to filaments, it pellets with them, right? So that's what Leah did. And she found that the FAB, which no one had ever tested whether it binds to actin, right? The FAB region accumulates in the pellet about, anywhere between 30, 40, 50%, right, depending on the experiment. It does that cooperatively with alpha-catenin, so it accumulates even a little bit better if you add alpha-catenin actin binding domain, right? And again, alpha-catenin actin binding domain, which is a pretty good actin binder, is only 50% in the pellet, right? So the FAB region is also a actin binding site. Kevin can use the new AlphaFold tools, right, to produce this predicted structure of that peptide bound to an actin filament, and particularly this helical turn, right, is well conserved between flies and mammals, right? And we think that's probably the actin binding site, and he's testing that. The conserved helices also bind, but they don't bind very well. But I believe that binding because Greg Aleutian's lab has documented that interaction by cryo-electron microscopy, right? So a beautiful paper from Gong in his lab, right? Brought together full-length alpha-catenin, full-length beta-catenin, full-length vinculin, and almost coincidentally, those two helical regions, if you pick that region, there were reasons, but that wasn't because of an AlphaFold prediction, because they started this before that, right? And what they found was the actin binding domain of alpha-catenin bound to actin like it should, except it bound with higher affinity when that ephedrine helical region was there. And this forked helix, which is touching both actin and alpha-ketinin, is strengthening that interaction. So again, I believe that this guy can also bind actin and probably alpha-ketinin. Okay, so we thought from this structure that this was going to be the key piece, right? This piece, because in their biochemical assays, strongly enhances interactions of alpha-ketinin and actin. But when we deleted the helical region, when we deleted the amino terminal spacer, or we deleted the C-terminal spacer, once again, we got viable flies. We did figure out where the nuclear export signal is in GeneMat, because the guy deleting the C-terminal spacer accumulates in nuclei, but it also accumulates at pretty much full levels in functions. And again, rescues to wild type. So going into the nucleus by itself does not mess up the function of this protein, right? And we also created a mutant where we only had the FAB and the conserved helical region. So we have both of those stickers, but neither spacer. And I'll just say, those guys are massively, they're actually worse than deleting the whole IDR. And that protein also goes into the nucleus, right? And then that says the spacers are collectively important, but are they individually important, and do they have sequences embedded in them? And Kevin, because he's my protein biochemist, he completely scrambled their sequence. They have the same amino acid composition, but they're not in the same order anymore. And we got a live fly. And it also goes into the nucleus, right? So that C-terminal region, there's a sequence in it that's important for nuclear export. So that's where we're at, right? And again, I have learned a lot, right? What I have learned is you really want to connect the junctions in the cytoskeleton effectively. And so you built in this tremendous excess of capacity, right? So that even though when I take this protein away, the whole system basically collapses in terms of junction cytoskeletal interactions. Almost none of its individual interactions are accepted, right? And so again, there's lots of questions, including whether it really is doing anything in the nucleus, right? But I'm happy, right? Because again, I got to take that machine apart, and for me, that's fun. And I got to work with some really tremendous people. Again, in that project, amazing graduate students starting at a postdoc who played an important role, but then all those postdocs, right, who just have really made my lab both productive and fun place to work. So thank you very much.
Time for questions.
You know what the nuclear export sequence is?
So, that they're good predictive algorithms for nuclear import sequences, and there are not. So if you use 5 different predictors, you get 5 different predictions. It's not a very long region, right? So, we could probably figure that out pretty straightforward with that way. But right now, with respect to that, my goal is to figure out, does it really need to go into the nucleus? And I will say, we already know a little bit. If I believe that the cat's tag sticks into the membrane, some of the flies carrying that is the only protein are alive to adulthood, but not nearly as many as if it's a wild type canoe. So that either means nucleolocalization, well, maybe the tag doesn't work, but if the tag works, nucleolocalization isn't essential. Maybe it's important, or maybe it's not really a very good idea to stick the C-terminus of this protein next to nothing, because that's what we're doing to the lab, right? We do have the GFP tag in between, so we'll learn there. But I think we could figure out where it is, and that probably would be useful. We do know where the NLSs are. They're up in the front near the RNAs. Yeah.
I was actually wondering about the analysis of data report.
Well, we haven't touched the short IDRs in the front half. So we haven't touched them, but there's more than one. So that's not going to be trivial, number one, and number two, it's going to be, you know, if I delete that region, I don't know what it's doing, right? So that probably is something we should eventually do, but I think that getting a straightforward answer to that's going to be a little bit more challenging. Yeah.
You mentioned that you know a little bit about how RAP1 is activating this guy. Is there any connection between that and the C-terminal domain? Or are they kind of working?
Okay, so first thing is, in my original view, right? This thing is folded into a closed conformation, right? And you can be familiar with that, right? And RAP1 binding opens up that conformation, right? Like formants, for example, right? That's how I originally imagined it. And that whole front set of 5 folded domains would be have a tertiary structure, right? Like inactive stalkers. But now I've deleted every single one of those domains. So I don't think that's true anymore. It's still possible that parts of the intrinsically disordered region fold back, right? And are important in that regulation. And but none of the individual domain deletions messes or individual region deletions messes that up. So what do we know? We know initially you need RAP1 binding to apically localize it, right, to where the junctions are. In the absence of the RA domains, it goes all along lateral membrane. In the absence of RAP1, it's not at the membrane at all. But as soon as gastrulation starts, it goes back. Because of this multivalence. But RAP1 is still clearly important for it, right? Because, you know, in the absence of RAP1, not only you formally apparently eliminate function, but you also eliminate the function of some other effector, at least one of which is tailoring. Right, so the RAP1 loss of function feature, that's actually worse than the phenotype. Again, a bunch of things that we thought it did, essential for membrane localization, it is essential for enrichment in response to force, but so is that C-terminal region that contains the nuclear export signal, but that fly lacking that is alive, right? So somebody wants to come to my lab and figure out what RAP1 does. Come on. Yeah.
With the deletion of that RAP domain, do you know what's recruiting it to the junctions when it's not there? Is it to the?
It's probably all of those pieces. It's probably the PBZ domain grabbing hold of the cat tail. It's probably the interaction with the enriched F components that are present there. The FHA domain and the dilute domain almost certainly bind partners. They do in the dilute domain in the unconventional myosin, which is the only other place it sits. FHA domains theoretically bind threonine phosphorylated peptides. Again, Kevin's mapped conservation. The dilute domain's got this big valley on the surface, is highly conserved. Something binds there. So we have no idea what binds to either of those two. What we do have is a very good set of proximity proteomics data on mammalian fadden that Longson showing my lab generated, right? So I've got a list of 100 proteins. It's probably one of them. Yeah.
I was wondering, because it seemed like a common theme with some of these mutants that you have in the canoe kind of structure function is that you have a decrease in the amount of canoe that's localizing to the junction. Is that right?
No.
No.
The only guy where that's obvious is deleting the entire proximal ADR.
Is there
What
any evidence?
does change again in the delta C, in the delta RAs, is the tension dependent enrichment at tri-summits. Right, but the junctional localization is not substantially reduced for any of them.
I was wondering in the Delta IDR case then, is there evidence that that region helps to recruit more canoe? Have you done?
That like it's does
Like
they
a
don't
sil-
make
like a
does they don't
silly
make
trans.
So again, we know the dilute domain dimerize. Kevin actually has some unpublished data that the FHA domain can also dimerize, right? So, but it's really, you gotta think about it, not like it's a network. Right? And so any one of those interactions probably might not be very high affinity, which put them all together, right? And then, you get fairly robust recruitment. We haven't done trap with canoe, but and somebody should, right? Because how often it's coming in and out of the complex, right, would be important data that we don't have. Yeah.
So I noticed myosin is not in your network diagram that's behind you right now. That's probably a big topic.
This is really interesting, and this is where I hope, right, this collaboration with your grandson is gonna help, because he's now working with Daniel St. Johnston doing DNA paint, and he's got 40 nanometer resolution. He can see the cadherin complexes on the two sides of the membrane, right? What we know at confocal resolution, you can see it in one of my images, right, is that I'm particularly interested in that time when where it's enriched in a planar polarized way at AP borders. What you see in the projection is it looks like it pulls away from the junctions, but myosin's still going to be cortical, right? It's not like flying out into the cytoplasm, right? So understanding the relationship with myosin is really interesting. Again, Lungson, this is now 10 years ago, right? We published some beautiful work that he did in this MDCK model where with Alan Fanning, we knocked down the ZO1 proteins for other reasons, right? That leads to really robust actin-myosin assembly of junctions in a sarcomeric array of myosin, right? And again, there we saw a similar thing, right? Where myosin appears to probably be spreading along the membrane. There, we could actually see myosin infiltrates, right? Which was incredible, right? And follow-up work from a different postdoc, Malaysia, UCAM, right? Saw that narrowing of an array of myosin filaments was also part of building that junctional network, which you took MDCK cells and did a calcium switch so that they zipped up, right? So I really want to know how myosin is connected to this. The other thing that came out of Jessica's work was when myosin moves, I think, along the apical membrane, right? PAR3 bazooka constricts away so that they don't overlap with one another. So there's some kind of, and again, Tony Harris has got some evidence of PAR3 and myosin are also talking to one another in some kind of mutually antagonistic way. But I really would like to know that. Yeah.
So for alpha catechin and F actin, you had the fact that they went and held it together with canoe, but what is the evidence for E-cadherin directly interacting with canoe?
That was a pull down experiment, right, with GST with the canoe tail added to it, right, pulling down a big chunk of canoe that we now know includes that H region, the C region, and the FAB. Yeah, that's work that Jessica Sawyer did somewhat heroically. That was the work that she got so frustrated and we went a little bit astray, and then she quit my lab for 2 days. But luckily, Bob Goldstein knocked her out of that, and she came back.
Back for one more question.
Okay, cool. Thanks.
All right,
That was really
thanks, Mark.
fun. Had a great day.